Okay, so yesterday, this should be where we finished up. We talked about the nucleus being the control center. It sent out information, basically the information to build proteins. Then we talked about the rough and the smooth endoplasmic reticulum, nicknamed the ER. And the difference between them is that the rough has little bumps on it, or little dots around it, which are what?
Ribosomes.
The ribosomes. And the smooth doesn't. Their jobs are very different. The rough prepares, modifies proteins, which makes sense because the ribosomes, which is the next slide, make proteins. So it makes sense that the ribosomes make the protein, and then right there next to them is the rough ER ready to modify the protein, just like an assembly line. The smooth ER doesn't do anything with protein. It has to do with working with lipids and carbohydrate metabolism. And I also mentioned yesterday that it detoxifies drugs. I think I mentioned that yesterday.
True.
And I mentioned about how, what organ would have a lot of them?
The liver.
The liver, good,
Yeah,
right.
you did mention that.
Because it's detoxifying alcohol, antibiotics. Pretty much that's the first place when you first absorb food from your digestive tract into your bloodstream. That's the first place it goes. It passes through the liver, where the liver is going to start to detox anything that you've absorbed that needs to be detoxified. It can only do it at a certain rate, which is why if you drink a lot of alcohol, it's going to keep circulating your system until the liver can get to it. But that's how that works. And then transport vesicles are just these little sacs that break off and basically take a product that's been made and carry it somewhere else. So it's just, you'll see examples of them on the next couple of slides. So, and if you didn't get any of this PowerPoint will be up today anyhow. All right, that's just a picture, which looks better up there, of the rough and the smooth ER. All right, so ribosomes. This is where I really wanted to pick up today. Ribosomes are made out of two things. rRNA. Do you remember what the little r stood for? Ribosome. So it's ribosomal RNA, and they're made out of a small amount of protein. Here's another question from yesterday. Do you remember what makes the rRNA? It's inside the nucleus. The nucleolus. Good. Inside the nucleus, it makes the rRNA that makes the ribosomes. They look like dots, but in reality, they're not dots. They're actually made out of two pieces. You'll learn more about this when we get to genetics, so you don't really have to know what the two pieces look like. But maybe if you remember your biology, you might remember this. There's a one piece, a bigger one, and a smaller one. And then I don't know if anybody remembers protein synthesis at all, but the mRNA was like this strand, and it had these three-letter codons like A, U, G, blah, 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 blah.
Yeah.
Sounds kind of familiar. Anyway, the ribosome, the two pieces are what hold the mRNA in place, so that when the protein's being made, it'll slide through here. If you don't remember protein synthesis, that's okay.
The U replaces the T, right?
Exactly. Yes. Good. All right. So they're made of two pieces. They can be free in the cytoplasm. Sometimes you have little ribosomes that are just sort of scattered. But you also have some that are attached to the rough ER.
This part is interruption. Did you write it?
So their job is to make proteins. The ones that are free-floating typically are making proteins that are going to stay in the cytoplasm. Cytosol is just another word for cytoplasm. The ones that are generally attached, meaning attached to the ER, are often making proteins that are then going to be modified and then exported to do something else, particularly in a multicellular organism. For example, we have certain cells that their job is to make a particular hormone. And most hormones are made out of protein. So that particular cell would have ribosomes that would then go, it would send the protein to the ER, that would modify it, and then it would get packed up to be shipped out of the cell to go somewhere else. A little sketch in the corner just showing the ribosomes that they're just little dots. The part that's going to be in yellow there at the bottom, you don't have to know for any test. That's why I highlighted it, just so you know. Golgi Apparatus, sometimes it depends on the book. It might be called the Golgi bodies or the Golgi complex. Those are just different names for the same thing. The word Golgi is always there though, so it doesn't really matter whether they have apparatus, Golgi bodies, Golgi complex. It just depends on the book, what terminology they use. So unlike the endoplasmic reticulum, the ER, which was long canals, this is going to look a little different than that. This looks like stacks of sacs. That's how I used to teach my honors class. Stacks of sacs. In other words, like this. Instead of being long canals, they would actually look kind of like this, like 4 or 5 stacked up on top of each other. Whereas, remember, the endoplasmic reticulum would look like this. It would just be like these really long canals, and you might see the dots around it as well, the ribosomes, depending what kind it was. All right. Their job is to modify and package products into vesicles for export. And here's the thing. Remember, the ER also modified stuff. What I really want you to know about the Golgi bodies is that they're like the UPS of the cell. They're going to make the final modifications and packaging to ship something out. So it is true that the ER can modify things, but this is where it's going to be the final packing place where it's going to get sent out of the cell. The stuff at the bottom here will not be on the test, and it shouldn't be on the AP exam either. The old AP exam maybe, but the one that we take now does not focus on little details like this. So it's here. You can copy it if you want to, and from Edline when I post this, but you won't have to know it for any test or for the AP exam. Cis side receives. The trans basically is the other side where stuff leaves, so stuff comes in on the cis side, leaves on the trans side. And in plants, sometimes they're called dictyosomes, which I have never once seen on a test anywhere. So I don't, it was one line in a book. I wrote it down, but you don't need to know it. All right, so don't worry about the stuff in yellow. If you want to copy it later, you can always get it online. And they can make polysaccharides as well. All right.
So wait, so are they monosaccharides?
They can put together monosaccharides into polysaccharides, meaning they can take sugars and then build them into starches. Yes? The Golgi apparatus can do that, yes. Here's a diagram. The green is an electron microscope picture of the Golgi bodies. The drawing at the bottom is just a representation. The little circles coming off the ends, those would be the vesicles that would be then carrying the product to some other location after it had been modified. All right, lysosomes. Lysosomes digest things. They have enzymes in them that will break down stuff. For example, if a cell was to eat a bacterium, you have cells that are designed just to eat and kill bacteria. What kind of cells do that?
Antibodies?
They make antibodies.
This is white.
Yeah, like T cells, white blood cells. All the different kinds of white blood cells have to do with breaking down viruses and bacteria. They also have a low pH. They have a pH of 5, which is slightly acidic. Remember we talked about how bacteria had just one big, large area. They didn't have any kind of compartments. So this is a compartment where specific things can be going on and allows these cells to be more complicated. You actually have some white blood cells that are, they are literally programmed that when there's an infection in the area, they burst their lysosomes and the whole cell explodes. They're like little Al-Qaeda cells.
So it's like.
So they're like suicide cells, literally. And basically that way it kills everything in the area. And that's what they're designed to do, to kill any kind of bacteria or infection in the whole area. They can either digest worn out cell parts. In other words, white blood cells, yes, they fight bacteria, they would have a lot of lysosomes. But other cells would have lysosomes too, because lysosomes also break down old cell parts. Right, autophagy, I think. You can write autophagy, but that sounds kind of wrong to me. And I also say phagocytosis. It sounds so much nicer than phagocytosis. So phagocytosis would be cell eating. It would literally eat something and digest it. It's in the background.
Hello.
All right. So auto means self. So autophagy would be breaking down, eating its own cell parts that are left over, that are old, that need to be recycled, and then the recycled bits can be rebuilt. And then phagocytosis, when a cell eats a virus or a bacteria or anything that doesn't belong. All right, peroxisomes are a little bit different. Just like they sound, they actually make peroxide, like the same stuff that comes in brown bottles that you can go buy at the drugstore, hydrogen peroxide. What happens is they break down fats. And when fats are broken down, one of the byproducts is peroxide. Peroxide itself is toxic. You don't want to drink peroxide. It is toxic to you. However, they make an enzyme, their own enzyme, to break the peroxide down. So in other words, what they really do is they break down lipids. A middle product of that is peroxide, but then the peroxide gets broken down. And if you've ever put peroxide on a cut, what does it do?
Burns.
Okay, besides stings and burns. It bubbles, yes. And the reason it bubbles is because of this. The enzyme catalase is actually in the cells around the area, and that's actually breaking down the peroxide into water and oxygen. If you had me for bio, we did one with liver, where we dropped liver in peroxide and it bubbled, and then we did the split test. You probably just remember burning wood. That was part of it. But we were testing for oxygen being made with the liver, because liver would have catalase in it. Which means peroxide, by the way, doesn't kill bacteria. So if you put peroxide on a cut, you're not killing anything. But doctors do use it to clean out wounds, because it does bubble out old skin cells, whatever. So a lot of times the first thing a doctor's going to do, put peroxide on, bubbles out debris or whatever, wipe that off, and then you can put on antibiotic ointment or whatever treatment that they're going to do to actually kill any remaining bacteria, just so you know. Okay, vacuoles. Vacuoles are basically for storage. In general, just vacuoles store things. In animals, there are small vacuoles. They're actually much more important in plants. In plants, the vacuole has a very important job. It stores water, and it's what makes the leaves stiff. When a plant wilts, it's actually wilting on the cell level. Remember, cells are the structural and functional units of multicellular organisms. So when a leaf is wilted, it's actually wilting on the cell level. In other words, here's our cell, and it's got a cell wall and a cell membrane. This is a plant cell. And normally, the vacuole is really swollen, and it's actually putting pressure on the cell wall. And if every cell in the leaf is doing that, now your leaf is stiff. But now let's say you don't water your plant, and water's being lost at the leaves. Some of it ends up evaporating. What's going to happen is this vacuole is now going to shrink. So now it doesn't really contain very much water. Now the cell membrane can actually pull away from the cell wall, and then the cell wall, it's strong, so it's not going to completely collapse, but what it will do is this. It'll kind of bow inward. And if you imagine every cell in the leaf doing the same thing, now you've got wilted leaves. So that's actually what vacuoles do in plants. They're very, very important in plants. Not as much in animals. Yes, they store things, but in plants, it's the whole reason why plants' leaves are stiff. Protists have contractile vacuoles. It's just a special vacuole that acts almost like a little bladder. What it literally does is, since they live in fresh water, water can constantly flow in. I don't know if you remember osmosis, that water goes from higher to lower concentrations. So an organism that lives in fresh water is actually going to have water tending to flow into it. What they do to compensate for that is, even though water's flowing in, they take their vacuole, and it acts almost like a bladder, and they just kind of squirt out excess water. And that way they can maintain a water balance. So it's not urine, but you can imagine it almost like a bladder, that it's getting rid of excess liquids. The most important one, though, would be to know that the vacuole is going to be really big in plants. If I was to show you a picture of a cell and I asked you if it was a plant cell or an animal cell, there's 3 things you would look for to identify it as a plant cell. Cell wall, the vacuole, chloroplast. Chlorophyll, yes, but if it's not a color picture, you might not know that. Chloroplast, which we're about to talk about next. All right, and I'm going to actually stop this here, and I'll get the remainder of it